Beyond the inference of gene regulatory networks, we also worked during last years in the inference of pathway networks. We started to develop this idea in cooperation with the Genomics of Gene Expression Lab at the Centro de Investigaciones Príncipe Felipe in Valencia. And we propose a new method that we call PANA. This method is oriented to study the functional interconnections about the molecular elements of the biological system. The idea with this method is work in two phases. In the first step, we make a transformation in the data. We try to define pathway activity profiles from gene expression profiles, making a transformation using principal component analysis. And when we finish this step, we have as an output a matrix where the rows are associated with the pathway activity profiles and the columns with the original experiments that we have in the gene expression data. And then we use Herner-Kopp to infer association and for extract association rules between these functional profiles. In this way, we achieve a network where the nodes are associated with the pathways and the links that connect the nodes represents crosstalk associations between the pathways. Here we have a summary of the methodology. We use as an input the gene expression data, and we take from KEGG the information about which genes belongs to each of the pathways. Using this information, we build sub-expression matrices, one matrix per each pathway. We use principal component analysis for extracting pathway signature. A pathway signature or a pathway activity profiles will be a principal component extracted from the different matrices. And the scores that we obtain from PCA method is used as an indicator of the activity level of the pathway. Using this matrix, we apply the machine learning method, we identified association rules, and from these association rules, we recover the structure of the network. Here I present a summary of the method. The method is more complex. For example, you can obtain from the method statistical information about which genes are generating the crosstalk between the pathways, and then you can use this information for analyzing the interpretability of the crosstalk detected by the method. We apply this approach for describing the functional transcriptional connections during the shared cell site using a microarray experiment oriented to study the cyclins in the schist. We also use this method to identify pathways that change their connectivity in a disease condition. In particular, we work with Alzheimer's datasets. And we also use simulated data in order to study the sensibility of the parameters that use this method. Well, this method achieve good performance, but it's also true that the method don't use all the information that you have available in terms of KEGG and in terms of the gene expression data. And for this reason, we define a new strategy The new strategy was oriented to incorporate information of the topology that we have associated to each pathway available in KEGG. And the idea of this methodology is, as a first step, we try to define a matrix that represents the complete chain topology of connections in the pathway pathways that we have in KEGG. And using this information, we apply a parsing method in order to detect which pathways are connected, topological connected. In general, we have three cases. We have the situation where two pathways have genes in common. We have the case where the pathways don't share genes but have connections between. There are connections between the genes of the pathways, and we have also the case where we don't have connection. Then the pathway topology matrix will be a binary matrix where we have the information about all the topological connections in the pathways, between the pathways. We also make an enrichment analysis from the microarray experiment data, and we detect which are the most relevant genes from this analysis. And then we assemble different matrices associated, one matrix associated for each of the links that we have in the pathway topology matrix. The idea, for example, when we are considering the link between pathway 1 and pathway 2, we will make a matrix, a gene expression matrix, where we only include the genes that belongs to these two pathways and are part of the list of relevant genes. And then we apply B-clustering in these matrices, applying the algorithm BGEA. From the B-clustering, we can analyze the degree of coverage that B-cluster have in the gene expression matrices. From this degree of coverage, we obtain a score, and this score is a measure of the intensity of the pathway crosstalk that we are detecting. And well, from this matrix, we can build the gene expression matrix, the pathway network. Well, we compare this method against PANA and PathNet, which is another method proposed for detecting pathways. In particular, we make a comparison trying to detecting pathways that change their connectivity in a disease condition using Alzheimer's datasets. In general, we found that PET, all the associations detected by PET are consistent with previous knowledge. We also achieve a significant coverage of the rules detected by PathNet and PANA. And we also detect some exclusive relationship with PET, and we analyze the information supported by the B-clustering analysis, and we found that the metrics associated with the B-clustering analysis correlate well with information that we have in ontologies. Well, in general, the experience with this method was very good. Well, as conclusions, several strategies for inference of biological network from gene expression data has been presented. In general, supervising methods as B-clustering can help to improve the gene expression analysis of the networks. Remain important challenges as the curation of the association, the curation of the crosstalk between pathway associations is a hard task. And we also want to work in the biological interpretability, give support for the biological interpretability of the network. In this sense, we are working now in the semantic analysis of network with HerenCode Plus, and we are developing text mining tools for helping in the curation of the methods for pathway crosstalk. The idea is try to search in the literature evidence that support the crosstalk interaction that you detect, and in a semi-automatic way try to help to the analysis in the study. And well, this is all. Thanks and questions.
In the beginning, you had the problem that you inferred that some gene 1 was regulating some gene 2, maybe with some time delay. Can you generalize this to some more complicated combinatorial processes, something like gene 1 and gene 2 regulate gene 3 with time delays T1, T3?
It's a good question. All the associations that we are detecting are pairwise associations. Okay, we always analyze associations between only two genes, but we are not considering, for example, regulator factors where you have a group of genes that need regulation on one gene.
Or some kind of I don't know, condition-dependent regulation. For instance, if you would use your B clustering and then on the clusters do the
when you are working. Sorry, maybe I don't. Oh. When you are working with the first method that I proposed, HerenCode, the analysis is pairwise, okay? But when you are working with the unsupervised method, the clustering, you detect a group of genes that are correlated, but you don't have direction in these associations. You know that all these genes are related, and the idea with let me show. The idea when you are working with this approach, where you combine HerenCode 2 with the clustering analysis, you have direction only in the associations, in the pairwise associations obtained by HerenCode. Okay? But when you decide to make an enrichment in the topology, selecting a group of genes and applying a clustering analysis, you obtain more associations, but these associations don't have direction. Okay? You need that this group of associations are related with